Hello, everyone. Um, so this is part one of chapter five, which are we're looking at the large biological molecules, or commonly called the macromolecules. So this here may not seem like anything to anyone, but this is actually a um, representation, the structural representation of alcohol dehydrogenase, which is one of the large molecules that exist in the body. Its job is basically to break down alcohol in the body. But there are tons of macromolecules that exist. And so let's go over several of the different ones um, uh, that we're going to cover in this chapter. Carbohydrates, proteins, nucleic acids, and the last group, lipids, are all of the four categories of macromolecules that we're going to go over and that you need to know about. So, macromolecules are polymers that are built from monomers. So, large polymers are known as macromolecules because they have a very large size. A polymer is a long molecule consisting of many similar or smaller building blocks. And we call these smaller building blocks monomers. Carbohydrates, proteins, and nucleic acids are all polymers that are built out of monomers. Now, when I think about monomers and polymers, I think the best explanation that I can use to describe the concept is that of like a pearl necklace. So this is a pearl necklace. And so if we imagine a pearl necklace to be a polymer, the individual monomers would be the individual pearls. So monomers are used to build and create polymers. And the ability to do this is through enzymes. So there are enzymes that are specialized macromolecules that speed up reactions and allow for the creation or breaking down of polymers. The process that is used to build polymers is called a dehydration reaction. It occurs when two monomers are bound together and there's a loss of a water molecule. You lose water, so you're being dehydrated. So it's a dehydration reaction. Polymers can be broken down and broken apart into the individual monomers by hydrolysis. It's a reaction that is essential for the reverse breakup of molecules. Hydrolysis is where you use water to cut um, molecules apart. So hydro meaning water, lysis meaning to cut. And so here's a representation of those. So in this first image here, this is dehydration reaction. So the bonding of the monomer, unlinked monomer to the short polymer here means that there is going to be a loss of water. So it's losing a water molecule and it's dehydrated. Hydrolysis is where we use water molecules. So we add water back into the system here. So water molecules can then be used to break it apart. And this goes back to the idea that water molecules are the universal solvents. So water molecules are able to do this quite easily. And so all of the macromolecules use dehydration and hydrolysis for the ability to create and break apart monomers of polymers. The diversity of polymers. So a cell has thousands of different macromolecules. Macromolecules vary among cells of an organism. They vary more within a species and vary even more between a species. Um, every cell is different. If we're talking about the human body, for example, different cells have different functions and jobs. And so Their ability to create and use polymers is vast. So the first macromolecule that we're going to cover is carbohydrates. Carbohydrates serve as fuel and the building material that is necessary for a lot of biological function. Carbohydrates include sugars and polymers of sugars. So if it is a sugar, it's a carbohydrate. If it's labeled as a fiber, for example, it's a carbohydrate. The simplest carbohydrates are monosaccharides, or what we call simple sugars. Now, the word monosaccharide. Mono means one, saccharide actually means sugar. So one sugar is the what that word means. Carbohydrate macromolecules are polysaccharides, meaning polymultiple sugars. And these polymers are composed of many sugar building blocks. Let's look at the sugars, monosaccharides. Monosaccharides have a molecular formula that is usually in multiples of C, H, and O. The best example is glucose. Glucose is the most common monosaccharide. It's one of the most common um, molecules on the planet. Monosaccharides are classified by either the location of their carbonyl group or the number of carbons in its carbon skeleton. So there's ways of identifying the monosaccharides. However, there are a lot of them. There's for example, the aldoses here and the ketoses, depending on the type of sugar. So you can be what they call it, trioses, meaning you have 3 carbons. Glyceraldehyde, for example, a pentose, meaning you have 5 carbons, like ribose and ribulose, which is very important for DNA, for example, or hexoses, which are uh, the most common here is like glucose and fructose. They're often drawn as linear skeletons. A lot of sugars will form rings. And monosaccharides are the major fuel for cells and is the raw material for building molecules. That is true for humans as well. Monosaccharides, for example, glucose is the major fuel for our cells. It's one of the reasons why we have to worry about blood sugar, and we'll cover that in a second. So sugars, monosaccharides can form uh, in different structures. They can be linear or they can be ring structures, which are seen here. Now, if I was to bind 2 monosaccharides together, it would be called a disaccharide, di meaning 2. So a disaccharide is 2 monosaccharides bound together. And once again, it's created through dehydration. This bond between the 2 monosaccharides is a covalent bond, and it's called a glycosidic linkage. Now, for a disaccharide it's not a monomer anymore. It's technically a polymer, but we have created a special group called the disaccharides because um, it includes one of the most important sugars that we consume on the planet, and that is table sugar. So you can see here in the image here, this is table sugar, uh, which is commonly called sucrose. So this is just showing you some examples of the creation of disaccharides. The 2 most common ones are maltose and sucrose. So for maltose, if you bound 2 glucose monomers together, you'll form maltose. For sucrose, if you bind a glucose and a fructose, you will create sucrose. Now, we can keep binding and adding monomers of monosaccharides together to form a polysaccharide, polymers. And polysaccharides can be very, very large. And they have several roles, including energy storage and for structural components. The architecture and function of a polysaccharide are determined by its sugar monomers and the positions of its glycosidic linkages. And this image here is showing you a lot of glucose molecules bound together to form a polymer. One of the storage polymers is a starch. Starch is a storage polymer of plants. It consists of long chains of glucose. So plants will store its surplus starch as granules within its cells, in the chloroplasts and other plastids of its cells. So plants will store uh, this starch to use later on as an energy storage. The simplest form of starch is what we call amylose, which is what we would find in potatoes. And so if we were to actually dry potatoes and grind them up, which you can see from this image here, you would see it's like a white powder. That is the starch. And then in this microscopic image, we can see granules of starch. These are starch granules. They're almost like little stones that are stored within the cells. Now, starch is a storage polysaccharide, and it's for energy storage. Animals are also able to do the same thing, and we don't store in the form of starch, we store it in the form of glycogen. So glycogen is the energy storage polysaccharide that are found in animals. Glycogen is mainly stored in the liver and in your muscle cells. And so in this image here, the blue dots represent um, granules of glycogen. So hydrolysis, or the breaking down of glycogen in these cells, releases glucose whenever there is a need for energy, whenever there's a need for sugar to be released into the blood. So this is just a review of the two different types of storage molecules between starch and glycogen, where one of the differences, starch is usually straight chains of glucose. A difference here in glycogen is that they are branched chains. They form branches, and it's a little bit more complex. There's another structural polysaccharide that plants will use, and that is cellulose. This is not for energy storage. This is structural. And like starch, it is very long, complex chains of glucose. However, there is a difference. Glucose chains will then bind together, and this is what this is showing you, to form fibers. So glucose chains will bind together to form fibers. And so once again, this is not for energy storage, this is for structure. And so plants will create cellulose to build the cell walls that form its body. So when you look at a plant, you're looking at mostly cellulose. And this image here is just showing you that there are different structures between the the monomers. They can be what we call alpha or beta monomers. And what's the purpose of that? I'm not really going to be testing you on alpha or beta configurations. What I do want you to know is that because is a difference in its chemical structure between, say, starch and cellulose, starch is easily digestible, where cellulose is not easily digestible. So some animals have evolved to be able to digest and break down these uh, cellulose structures. For example, like cows, herbivores like termites and cows, and they're able to break down these cellular structures where some animals are not able to do that, like humans. A lot of humans, we are not able to digest or break down a lot of cellulose. And so we rely on other things to help break that down, or it just passes straight through your digestive tract. And so we call those things insoluble fiber. and because all it does is just goes through the digestive tract. Chitin is another structural polysaccharide that's not found in plants, it's found in animals. From the exoskeleton of arthropods um, to, for example, in fungi. And so chitin is used for structural support for cell walls of many different things, or it's used to build exoskeletons. And so this is showing you the image here, an exoskeleton of some sort of insect here. I don't think it's a cicada, but it's very similar, but I don't think it is. Anyway, chitin is a structural polysaccharide that is seen in animals. So let's go over some functions of carbohydrates just to review um, some of the functions of these and put importance of carbohydrates. The number one function of carbohydrates is energy. Energy is stored either as starches for like in plants or glycogen is in animals. Plants in particularly store a lot of energy in the form of carbohydrates. And so humans, we eat those plants. So plants will store a lot of energy in its seeds. This is wheat in the image. And we love wheat as humans. We will harvest wheat and create our breads, for example. And plants store a lot of energy uh, in the form of starch. Glucose is the carbohydrate of most importance to living organisms. Most sugars are digested and converted or broken down into glucose. Glucose is what we use for our cells, it's the fuel for our cells. And so when we have too much glucose, we store it as glycogen in our liver. And if we have too much glycogen, we convert it to fat. Glucose is also the sugar that's responsible for your blood sugar. And so it's important to, for some individuals, we're not able to um, process sugar. Either you have too much sugar or too little. And so it's important that we have to keep track of that. Uh, Carbohydrates are also important in carb loading, right? For energy. Athletes would deplete their carb stores in their liver, and then right before a competition, they will carb load, where they'll eat a whole lot of carbs to give them a boost of energy right before a competition. Carbohydrates actually has a second effect in that um, it also helps maintain water weight, which can be good or bad. So water is a polar molecule, and so it likes glucose. And so reducing your carbohydrate intake can help reduce your water retention. So at holidays, whenever you're always eating a little bit more, you also all gain a lot of water weight. The second function of carbohydrates is structure. So chitin and cellulose are the examples we've covered about structure. Structural carbohydrates are actually involved in the physical makeup of the organism. For example, chitin makes up exoskeletons in insects and crustaceans, and cellulose is the cell wall material that's found in plants. And so here in this image, we can see some examples of organisms and chitin. Now, this image here, this is a cicada that is growing out of its exoskeleton. But not only do insects form their exoskeletons out of chitin, so do crustaceans like crabs, or the only land crustacean, the roly-poly. Fungi also fill their cell walls with chitin, which is why if you've ever felt or grabbed a mushroom, it's a little spongy, and it's because of that chitin in its cell walls that it is spongy. And then the last was cellulose. Cellulose is what helps build plant cell walls. And these images are just showing you how cellulose is used to lay down like scaffolding to build the actual cell walls of plants. And with that, that finishes carbohydrates. In the next video, the next part, we're going to review how to um, the other macromolecules, in particular lipids.